How's it going, Future Cannabis Project community? Back again, it's that rotten son of a blunt, Lobster Fam Farms, joined by our preeminent biophysicist and photosynthesis, Nick Nikolaev from Rooted Leaf. And Josh from YellowHammer is here to join us as well, as long as the big boss Nuss, Peter Cerveri. And today we are going to be talking about sugar phosphates and their role in photosynthesis and plant expression, among some other interesting topics. And from what I got on the pregame, this is some very under talked about and underappreciated, but very interesting actions that are going on. And I'm stoked to learn something new today with everyone. One thing I wanted to say too, it's been about a year since Nick's generation 3 of his product, the V3 of Rooted Leaf, has been around. And I guess maybe that's a good place to start, is kind of what have you noticed in the last year in the new product formulation?
Yeah, it's great to see you, Travis. It's great to meet you, Josh and Peter. I guess it's okay to see you too. I'm just kidding. It's always great to see you, and I appreciate the opportunity to come on to FCP yet again. It's been a very long time. I miss everybody that is watching, so thank you guys for tuning in. We've been rolling with Gen 3 for a little bit over a year now, and so we've had a chance to collect a lot of feedback from people and see what are some of the changes that they're noticing in the garden, the activity of the plants, and we've had a chance to kind of correlate a bunch of stuff together. So now I can kind of more confidently speak about some of the results, and we can hopefully treat this as an opportunity to start peeling back some of the layers about, what it is we're doing now with Gen 3 that's a little bit different, and taking a look at it from the perspective of that I hope we can shed light on some of these topics in a way that gives people inspiration to think about the future of fertilizer technology, because it seems like there's just so many advances coming out and people are learning so rapidly and so fast about botany and biochemistry and plant biology. But on the back end, how we manufacture fertilizer hasn't really changed significantly in a very long time. So I hope that people start to look at opportunities and potentials for advancing fertilizer technology forward. And a lot of it has to do with some of the stuff that we'll be, talking about today as far as sugar phosphates go. I'm hoping I can kind of lay the groundwork for people to look at sugar phosphates as the new sort of PK booster, if you will, or the future of phosphorus fertilizers. But in order to do that, we'll have to lay a little bit of groundwork first. So I kind of want to treat this as an opportunity to roll out some concepts about plant biology that are a little bit new, but that don't necessarily disconnect people from what they're currently used to thinking about. Sugar phosphates feel like a really natural place to start because that's what plants produce through photosynthesis. The products of photosynthesis, as we would refer to them, are sugars, but more accurately, they're sugar phosphates. And so when we start to organize plant activity based around the various flavors of sugar that they're working with, we start to see that we can organize it in this high-level understanding. I refer to it as a sugar phosphate spine, if you will, because you start off with a basic 3-carbon sugar, which is what C3 plants like cannabis plants will produce through photosynthesis. And then you've got one node above that, which is a 4-carbon sugar, and one node above that, that's a 5-carbon sugar, and one node above that, that's a 6-carbon sugar. It does go higher than that, but I think for the purpose of this discussion, keeping it in that window of 3 to 6, we'll also include 2-carbon, something about 2 carbons here in a couple of minutes. But the idea basically is that this 3-carbon sugar is really important because what it does is it represents a stable energy state that the plant has access to. If you think about what it takes to make that sugar into a stable form that the plants can translocate and move around and access and oxidize and break down and release energy and fuel their own growth with, that comes from the electron transport chain. That comes from ATP being generated, also as product of electron transport and the function of the oxygen evolution complex. And then we have CO2 released to rubisco to power its carboxylation activity. These things have to come together. The electron transport chain that deals with electrons, the proton side of the equation that generates ATP and fuels the release of CO2 to rubisco, and the actual CO2 itself, those have to come together to make this sugar. So the sugar itself represents something that's a fundamentally stable state for the plants. From this 3-carbon sugar, they can then work their way up and produce more complex molecules. And the way that I like to think about it, something that helps me personally is, we've got this vertical axis for just the C3, C4, C5, and C6 sugars that we're working with here. Those go up and down, just vertically, straight up and down. And then along each node point, we have these branches that come out horizontally. And these branches are the things that inform downstream metabolic activity. So if we're looking at horizontally outward from C3, it's really anything that a plant can do and everything that a plant can do, because that's where it starts from. That's what photosynthesis is. It produces a 3-carbon sugar phosphate that becomes the basis for all of plant biology. If we go up one node from there and we look at 4-carbon sugar chemistry, we start to realize that this specific node is prepared very well for terpene biosynthesis, specifically plastidiol terpene biosynthesis. In fact, the name of the pathway itself is methyl erythritol phosphate pathway, MEP. The erythros is the 4-carbon sugar, and the phosphate is the fun bit that we'll end up talking about, the sugar phosphate bit. But the point I'm still trying to establish right now is that when we look at where this 4-carbon sugar erythros or erythros phosphate pathways in general tend to branch out to in nature, what we find is that a lot of them lead to terpenes. In fact, predominantly it's the 4-carbon sugar pathways that will lead to terpene biosynthesis, particularly true for cannabis plants. There's over 70,000 different terpenes that are produced in nature, and to think that we might be able to organize them all as being traceable back to one node on our little sugar phosphate spine is actually a really good way to organize an otherwise very large and diverse class of metabolites down to a central origin point that they all have to pull from. They're all pulling carbon substrates down this one pathway. So the C4 node becomes very, very important. C5 node is also very important too, specifically for cannabinoid biosynthesis. And as it turns out, some of the latest and greatest scientific data out there about the function of cannabis trichome heads and the role that certain enzymes play and what are the major expressed pathways in trichome heads. The data that's coming out supports this idea that, the pentose phosphate pathway in plants is extremely important. Pentose phosphate pathway in cannabis plants is extremely important for the biosynthesis of cannabinoids. And pentose, of course, 5-carbon sugar that the plants are dealing with. So it becomes very important for us to understand when we're talking about these complex metabolites, it's not just like the plants produce sugar and then the sugar somehow becomes a cannabinoid. There are these intermediary nodes where the plant is building up, the 3-carbon, the 4-carbon, the 5-carbon, and even the 6-carbon sugars, and then using those intelligently in a way to branch out into all these terminal metabolite compounds. In a lot of ways, along the way, there's plenty of crosstalk between these pathways. One of the best examples is the cannabinoid molecule itself, because there's 2 distinct pathways that have to be activated and expressed in order to make a cannabinoid. One of them is the polyketide pathway, which has nothing to do with terpenes, and the other is the terpene pathway, which has nothing to do with polyketides. So in a lot of ways, actually, these 2 pathways are really fun to talk about because polyketides, generally speaking, in terms of biochemistry, they happen to be very neat and clean and organized. They're almost template driven in the sense that you've got a very specific enzyme that contributes a very specific piece through a very specific reaction that's well characterized. And the stuff is very modular. It's very template driven. So you can accurately predict, when you're modeling the function of some of these polyketide synthase enzymes, you can model their function very effectively and very accurately. Because it's template driven, you can kind of predict the outcome a lot more efficiently than you can with something like terpene biosynthesis, which happens to be promiscuous in a lot of ways. I think we talked about that in one of the episodes in the past, but the idea is that terpene biosynthesis is a little bit like the Wild West in terms of substrates going in and products being produced, and there's maybe a little bit less overall continuity like we would find with the polyketides. But these 2 pathways have to converge together to create CBG, which is the first progenitor cannabinoid that's in the equation. And in order to talk about polyketides, we actually need to give a shout out to acetates or acetyl coenzyme A, because it's that C2 metabolism specifically that fuels the production of ketones. And so if you're feeding things like low molecular weight organic acids to your plants, some kind of maybe an acetate mineral complex or something like that, you're literally contributing directly a building block, a substrate that actually contributes to the formation of a cannabinoid downstream. So what you put in as far as an acetate mineral complex initially may end up actually contributing to the final biomass of what's present inside of, a cannabis trichome head at the end of the yield. So The last flavor that I want to talk about before I get off the stage here, because I'm talking way too much at this point, but the last one is a 6-carbon sugar, and the reason that it's relevant in cannabis plants specifically, well, there's a couple of reasons, I guess. One of them is that it does play a role in some of these 6-carbon sugars, like inositol, play a role as osmoprotectants in the actual matrix of the cavity itself, the trichome head. But there's also some really interesting biochemical properties that inositol has, like plants can convert it into vitamin C, for instance. And so for dealing with basic redox stress, this is an example of a kind of a sugar, if you will. We'll trace it out. It's a kind of sugar that can be converted into vitamin C for the plants to be able to use. So it's profound to what extent plants are able to take some of these different nodes of the sugar phosphates, C3, C4, C5, and C6, and branch them out into everything that we think we know about cannabis plants in general. Like all the terpenes that are produced, all the cannabinoids that are produced, really all of the metabolites that are produced are possible for us to trace back to one of these nodes that exist between C3, C4, C5, and C6. And I will say real quick, as kind of a final wrap-up point, even if you grow for seed, the type of phosphorus that's stored inside of the seed is inositol phosphate, IP6. This is the type of phosphorus that the next generation of plants will be able to access and use to grow. So initially, just to kickstart the growth and to get things going, they rely on this stored form of phosphorus that happens to be a 6-carbon sugar phosphate. It is the fuel for the next generation. That's how important it is. And with that being said,
that's interesting you brought that up, Nick, because I've used this compound in embryo recovery for old seeds that I basically do embryonic rescue on rare or old seeds, and in addition to some enzymes, this phosphate compound is absolutely vital to bringing old seeds back to life.
Yeah, it's interesting that you say that because, like I mentioned with cannabis plants, the natural biosynthesis, the pathways lead to IP6 as a storage form of phosphate for the next generation to be able to access. So it's interesting that you're getting those kinds of results, feeding it exogenously to the plants.
Yeah, I mean, it's really, I think one of the things you talked a bit about terpene and cannabinoid expression, and as somebody who has a background in biochemistry and plant physiology, essentially terpene and cannabinoid expression is fundamentally a carbon partitioning problem, right? And what you're talking about with sugar phosphates, those are the currency of carbon allocation, right? Like without sugar phosphates, it's kind of a sink source issue, but photosynthesis essentially stops, right? The pathways can't move forward without these compounds.
Yeah, and from what I understand, some of the literature that supports how mycorrhizal fungi can increase phosphorus nutrition status in plants, regardless of the actual element being increased, is by helping regenerate the acceptor that rubisco uses in its carboxylation activities, so that ribulose phosphate sugar itself becomes regenerated or recharged when you have the influence of mycorrhizal fungi. And I think this kind of it's in the same nexus of influence, because when plants are working with phosphorus as an energy currency in the form of ATP, there's a lot that goes into that. It's not just like the phosphorus exists, or the phosphate group is kind of like detached from everything else around it. You have to suspend that in an organic environment and put it in its native field, I guess you could say, because field physics is very important in biology. Particularly with the solubilization of CO2. It's one of the topics that I like to kind of get into, and hopefully we'll branch off into that. But yeah, the sugar phosphate thing, from my perspective as a fertilizer manufacturer, is this really does represent, the ability to feed specific sugar phosphates to plants represents what I perceive to be the future of phosphorus nutrition, because there's this shortcut we're taking to the energy expenditure required and also inserting the sugar phosphate further downstream than just where the Calvin cycle ends. We're talking about way downstream at potentially pathways where it can quickly be introduced and then lead to a branching, set of pathways from there without having to go through any kind of metabolic bottlenecks.
Yeah, and this is really important from a cultivation standpoint because, what sugar phosphates actually are, things like G6P, glucose 6 phosphate, F6P, fructose 6 phosphate, G3P, which you mentioned, glyceraldehyde 3 phosphate. You mentioned ribulose, like ribulose 1,5 bisphosphate. Those are activated carbon forms. They're not storage. They're not structural in the sense of anatomically the plant. They're essentially like decision nodes, forks in the road that determine metabolism. And these phosphates eventually determined where fixed carbon goes. Does it go to growth? Does it go to storage for stress events? Or does it end up in our metabolites like terpenes or cannabinoids?
Yeah, and in some cases, maybe it's a function of both, because I'm, fairly convinced that glycerol ends up as a major constituent of the actual trichome structure itself, the trichome head. Plus, you're likely to find inositol in measurable concentrations as well. So these could, at some point, come close to the total terpene concentration on flower. So I'm talking about 2 to 3% by weight could be glycerol, could be maybe a little bit less inositol, but certainly glycerol, because it's a triol. Yeah. Sorry, what were we saying?
That's significant.
It is, yeah. And if you think about the environment that a trichome head represents, like, in many ways, we were maybe talking about anthocyanins recently in a discussion, and I was kind of comparing and contrasting the environment of a vacuole with that of a trichome head, and how one is filled with water and it's full of water-soluble components, and kind of like wraps itself around the cell and kind of, does this weird fluid thing. But the trichome head itself is not that at all. It's kind of like the polar opposite, and yet in some cases we see anthocyanins themselves bleeding in through either through the stalks. There's not really strong evidence that suggests there's anthocyanin synthase enzymes in the actual disc cells, or they're not even present in the actual heads. So the idea maybe is that you've got this major flux of metabolites coming in from the flower, and some of that could be phenolic compounds like flavonoids that are direct precursors for anthocyanins that just end up, there's some crazy chemistry that happens, and they just end up being fully synthesized in the stalks and in the heads. We got a little off track there from sugar phosphates. I apologize. What were we talking about there?
Well, I think we're just talking a little bit about how these sugar phosphates are so important in, determining overall outcomes of
like it's
carbon.
a major limiting factor. I feel is like kind of where you guys were going, like, maybe the limiting factor. Seems like it's almost the genesis of everything in the plant, which to me is like a really cool way to look at it. It's like, if you're saying everything that humans really care about the plant as far as like consumption, maybe not like rope and all the other nutrition, the other things like seeds, but for smoking and getting aromas and flavors and good buzzes, it seems like it's the crucial genesis of all that.
Yeah, and to the larger point about, like Josh was saying earlier, cannabinoid biosynthesis is largely a function of energy and substrate flux. That's why you need high light intensity, and you need high amounts of carbon, typically in the form of CO2. But if you can figure out how to put carbon in the water in a chemically reduced form, maybe there's some energetic advantage there as well. But yeah, so if you can increase the rate at which plants are able to access some of the otherwise rate limiting substrates, and they can quickly convert it into compounds that represent high energy sinks, like terpenes and cannabinoids are major sinks for ATP and for reduction power in NADPH. That reduction power and that ATP quite literally can be represented by these molecules. So the idea basically is that if you can somehow substitute the energy load that's required by feeding plants forms of these sugar phosphates that are native to their biology and plug in along these nodes specifically, and allow for the downstream expression of things like whatever is downstream from MEP pathway in the plastids, or whatever's downstream from pentose phosphate pathway in trichome heads, or on the flip side, whatever cannabis plants are doing with inositol phosphate, either storing it in the seeds or putting it into the trichome heads. If you can increase the rate at which all of that stuff is done, you get different expressions from the plants. They're not passive viewers to this type of stuff flowing through their systems. They actively respond to these things. And I think we see that as certainly being true when we're talking about biostimulants, like with kelp extracts, for example. There's some signaling stuff going on where the plants pick up whatever's being put down via that solution. It's not necessarily like the kelp is delivering this massive carbon load, and this carbon is like the raw material to make more terpenes and to make more cannabinoids. It never approaches that level of feeding the plants enough, but there's a signaling thing that's worth kind of peeling back, and talking about a little bit in greater detail. I mean, if you guys want.
Absolutely. So I'm thinking about it a little more. Would you guys kind of say that if the sugar phosphate is the end result of photosynthesis, besides, let's say, as nutrition, is that kind of the limiting factor? Like, let's take light and temperature and all the other outside stuff. Would that be?
No.
Yeah, I mean, yeah. Well, you go ahead.
That was awesome. And no and a yeah at the same time. Love it. This is perfect.
Well, I was speaking more specifically about the environmental factors, not necessarily like the sugar phosphate. Sorry.
Yeah, no, I was going to say that there's, aside from having the substrate, you need the reduction power to afford those chemical processes to actually occur. So it's almost like you have two components of the equation that sugar phosphates wrap up nicely into, because they happen to feed both. They can deliver the carbon that becomes the actual cannabinoid, but they can also release the energy that powers the process of making these cannabinoids themselves. So that's why they pose a distinct advantage relative to some of the other things that we might be looking at or we might be talking about.
What systems do you guys think are lacking the most? Like, I don't know how to really formulate the question. Like, is there a certain style of growing that could really be inhibiting these processes, nutrition-wise, maybe, or like?
Yeah, I mean, from my perspective, I think feeding fully oxidized forms of nutrients like a purely salt-based fertilizer oftentimes changes the redox landscape that plants have to navigate, because everything that they're taking up in the feed water requires reduction power to be able to effectively access. So they end up establishing this sort of electrochemical gradient, if you will, on a organism-wide level, where most of the electron transport chain activities are associated with things that revolve around conversion of inorganic salt-based elements into organic forms. You can take little shortcuts along the way, like feeding amino acids instead of pure nitrates, and there's some benefits there along redox chemistry and along having carbon as a substrate available for plants to do things like make structural proteins or just like make functional enzymes and things along those lines. I would say that's the biggest thing from my perspective. Yeah, Josh?
Well, I was just going to say, kind of the ceiling on the plant chemistry and a lot of the chemistry you're talking about kind of starts with how large and how mobile that this sugar phosphate pool is. And, kind of going back to the sugar phosphates, like plants need this pool to be able to modulate their metabolic function in response to the environmental cues and the genes that are either turned on, turned off, or switching on, switching off at any given time. But I like what you're talking about with essentially taking metabolic shortcuts. Instead of forcing the plants to do all of this work to convert, oxidized forms or reduced forms of, say, phosphorus or calcium or whatever nutrient, basically just give it to it in a way that you cut out this whole step, which essentially is, saving an enormous amount of energy for the plant and allowing the plant to devote that energy to other processes.
Yeah, and that's where we find more of the energy becomes available for things like gene expression. And I wonder to what extent the landscape to navigate isn't related to signaling like electrochemistry more than substrate availability, because there is something to be said about, for those of you that are familiar with the way that enzymes work and substrates work, it's like, generally speaking, enzymes are surrounded by their substrate, and at a certain point of being surrounded by their substrate, they kind of get the flip switch to turn on. So effectively, you've got this enzyme substrate complex, and you've got this environment where the enzyme can flip on and run for a long period of time without fully depleting its substrate. And this does a lot of very interesting things in nature, like in pollen, for example. In pollen tubes, you may have some enzymes that are active. They're actively suspended in their substrate, which is a sugar, or maybe it's like sugars, and there might be some like glycerol and polyols that are in the mix too. But you've got this enzyme that's suspended in its substrate, forms a complex, the enzyme turns on, and it starts transforming the substrate around it into a product that could serve as an attractant to attract a hummingbird. It's not so much that the sucrose content of the flower is attractive. It's some other compound that's produced as a result of an enzyme that's suspended in the sugar and converting the sugar into this attractant that brings the hummingbird close, or it attracts it, I should say. And, the hummingbird does its thing. So we have to think about how gene expression can also take on, like, we have to look at it from a different perspective. If we can saturate the plants with substrates that are required for the expression of terpenes and cannabinoids, can we put them in an environment where they get the on switch flipped a lot sooner than they otherwise would? If they're relying on pure salt-based fertilizers and they're relying on the Calvin cycle to reduce CO2 out of the air into a 3-carbon sugar, G3P, that's going to take some time. But if we can introduce, between the CO2 in the air and the cannabinoid that's inside of your trichome head, there's a large amount of time and a large amount of different steps and different complexes, different pathways, different enzymes have to get involved. There's all this convergence and crosstalk that exists. So if we can somehow take a shortcut and bring something to the plant that's already closer to the final form, that could benefit us in a lot more ways than just us seeing an increase in the concentration of cannabinoids and the concentration of terpenes. We also see an increase in the depth and the breadth of the expression. We see different terpenes that maybe typically are a little bit more rare or conserved that are coming out kind of front and center. I had a pineapple upside down cake that tested at 3.5% eucalyptol, and I was like, eucalyptol 3.5%. Alpha farnesene, beta farnesene, cis farnesene. The farnesene fraction tests super high with, some of the flower that's grown with fruited leaf. So we see a difference in the terpene expression overall. It's not just like we're getting more myrcene and we're getting more linalool and we're getting more beta caryophyllene and more pinene, right? These are the usual suspects. It's more about the exotic ones that typically don't come out to such a high extent. That's the interesting part for me.
Yeah, that's cool. That kind of ties into the conversation that we were having before, like chemovar expressions due to nutrition or other impacts. That's pretty cool to hear that you could have such a drastic impact, almost seemingly make terpenes appear that like weren't really there before. Like that's serious.
Yeah, and I don't pretend to fully understand the biosynthetic logic. Did I say that right? Biosynthetic logic, yeah, behind it. So eucalyptol, pineapple upside down cake. I forget the breeder. I'm terrible with breeders, by the way. I couldn't tell you who bred what.
Do you remember what substrate he's asking as well? If it was like
Promix.
Promix?
Yeah, okay. Promix HP. And this was grown in just south of Norman, Oklahoma. Yeah.
Too cool.
Yeah. So, I mean, I don't, terpene biosynthesis is this very kind of interesting, convoluted, not convoluted. It's interesting because there's promiscuity to it. And like the way that I like to think about cannabinoids is that you have two parents of this pathway, one of which is kind of like for lack of a better phrase, very loosey-goosey, just kind of like makes up their own schedule, in the moment, in real time. And the other parent is like this very clean cut, very organized individual that just gets everything done right on time. So the polyketide pathway and the terpene pathway are really like two polar opposite examples of biosynthetic logic in plant biochemistry. And yet we have cannabis plants that are making these terpeno-phenolic compounds and converging them together in this beautiful molecule that we call CBG. Eventually it becomes differentiated into THC. And decarboxylated, of course, into THC. But the point is, the cannabis plants are doing things that are quite remarkable in terms of the biochemical considerations required when we're talking about merging two different pathways with such vastly different fundamental logic that guides the process of producing these terminal metabolites. And the fact that they converge so neatly and cleanly is quite remarkable in cannabis plants, and at such a high concentration too, because we're not just talking about 1 or 2% total cannabinoids being present here. We're talking about 25-30+% of the weight of these plants is in the form of a cannabinoid. So to your point earlier, Josh, you were saying that availability of carbon as a substrate and then just energy flux in general is going to be the limiting factor for the biosynthesis of terpenes and cannabinoids. And to some extent, it might be possible for us to quickly do some math and try to figure out like how much carbon is associated with those cannabinoids in the flower. That could actually be really quick, fun, and easy to do, because cannabinoids are roughly 80% carbon by weight. So if I give you a pound of flour that's 25%, tested at 25% total cannabinoids, it's the same thing as saying the flour tested at a total of somewhere around 20% carbon just from the cannabinoids. If we have 3 to 5% terpenes on that dry flower, the monoterpenes and sesquiterpenes that are oftentimes found in cannabis plants, those are about 90% carbon by weight. It's like 88 point some odd percent, but we'll round up and we'll say it's 90% carbon by weight. By the time we're done adding up the amount of carbon that contributes to our total cannabinoids and our total terpene fractions, the amount of carbon ends up being at least a full, quarter of a pound of the flour, if not greater, a third of a pound of the flour, which is nuts. You wouldn't look at your flour. You wouldn't look at a pound of flour and think that 1/3 of what I'm looking at is just one element that's wrapped up in all these different molecular configurations in a way that gives the illusion and the impression that the leaf is somehow distinctly different from the trichome head and the stalk, and all that is somehow distinctly different from the contents of the head, which is the cannabinoids and the terpenes. But if we peel it back far enough, we realize it's all carbon, all of it. The predominant majority of it is carbon. We're dealing with stuff that's mostly carbon by weight. So figuring out how to increase the throughput of carbon by feeding sugar phosphates, I think, is one of the best things that people can start to think about and try to process mentally.
Yeah. One thing too, talking about that and feeding the sugar phosphates and talking about carbon, and it's, majority component of these compounds. One of the things that I see frequently is a overdosing of nitrogen later in the flowering stage, which leads to more amino acid and protein synthesis and vegetative style growth, which competes directly with that carbon allocation to those secondary metabolites. So, setting your nitrogen feed to ramp down and reducing it gradually as you enter during flower, it's really important if you want to drive the carbon allocation to the secondary metabolites versus structural components.
Yeah, there's definitely crosstalk competition between reduction power. Like the Calvin cycle itself requires the reduction power that's generated from the same electron transport chain that also feeds nitrate reductase in the leaves. And, nitrates can be taken up in high concentrations in plants without burning tissue. They're actually pretty benign overall, but the issue comes down to how much reduction power is required to convert one of those nitrates into an amino acid form. And then from there, you still have to solve for the question of where does the reduced carbon skeleton to situate on top of that ammoniacal head group, where does that come from? So you almost like need this biosynthetic logic that speaks to understanding that nitrate reduction and carbon reduction are competing for reduction power, but at the same time, they have to converge together in order to actually make an amino acid. And I guess one layer on top of that would be to look at cysteine specifically as an amino acid, because there we're adding a third layer, which is sulfate reduction. Now we need to talk about sulfate being reduced into a thiol group that becomes a constituent of cysteine. So we have sulfate reduction, nitrate reduction, and carbon reduction that all have to converge into this amino acid that we call cysteine. So the biosynthetic logic that plants follow to make cysteine is probably one of the most beautiful in all of nature, in my opinion, because it's this convergent point of three different things that otherwise compete against each other. I think one of the strategies, to your point about reducing the overall nitrogen load, that seems to help some growers is changing the nitrate to ammoniacal nitrogen ratio just a little bit in kind of like the mid stages of bloom, because nitrates will tend to promote more stretching, so you get greater internode spacing, generally speaking. It's not a purely nutritional driven phenomena, of course, but you could feed amino acids to plants, and that encourages maybe a little bit less internode spacing, more tighter internode spacing, so the plants don't stretch out as much. And by changing the profile very slightly, if you maybe start off at 80 to 90% nitrate and bring that down to about 66 to 75% nitrate and try to feed more aminos at very specific times in the plant's life, you can do certain things like keep the plants from stretching too far. If you're running some genetics that will at least double in height, and you're like, man, this plant's already 36 inches tall, like I just don't have the ceiling space to do this. One of the strategies might be to try to tighten up that nitrate to ammoniacal nitrogen ratio and try to feed the plants a little more aminos. A little more silica can help keep node spacing tight too, although that's for different reasons aside from nitrogen metabolism. But yeah, overfeeding nitrogen is a big one. Overfeeding phosphorus, PK boosters used to be a huge problem, I think, because people didn't know what they were handling. And now the latest craze is that people are overfeeding sulfur by a freaking significant amount, by a huge amount, because of that paper, that beautiful paper, by the way, that came out, I think 2019, or maybe a little bit more recently, that had looked at the volatile sulfur compounds that were present inside of cannabis trichome heads, and it quantified them in the parts per trillion. And I think people got the wrong impression about, feeding Epsom salts to their plants as a direct result of that. So it seems to come and go in waves, these ideas or these philosophies about what is it that's going to work to get my plants that roadkill skunk profile, and like putting Epsom salt on your plants isn't really like isn't the hardcore throughput way to just force your plants to do that. There's more nuance, there's more biochemistry. It's not just a function of, is there enough sulfate present or not? So
total side note, but I actually did have a chemistry question for roadkill skunk since you brought it up. So are alkaloids aromatic, and could it be alkaloids that are responsible for skunk blood? And would alkaloids be what's responsible for cocaine's aroma? Now, here's why I ask, because I smelled some rosin that was made from the roadkill skunk, and it was identical to cocaine. And my mind has been, my bro science mind, not my educated chemistry mind, my bro science mind has gone to, okay, maybe it's the alkaloids in coke that are giving me this aroma, and somehow the alkaloids are coming out of the roadkill skunk. They captured it fresh frozen, and I don't know where my mind's going, but to me, that could be the missing link of potentially of where the roadkill skunk aroma is coming from, and how do we coax more alkaloids if these are aromatic? Total blind speculation here, but that's where my mind's at on the roadkill.
Yeah, I mean, aromatic in terms of being able to smell them and detect them with your olfactory senses, or aromatic in terms of chemistry. Do they have an aromatic ring group inside of them? I think what he was talking about was the aroma that you could detect, like the odor. I don't know what cocaine smells like, to be perfectly honest.
Okay. Like gasoline, like real fuel. Like, I don't know how else to describe it, but like rocket fuel, like highly refined fuel.
Without knowing, so I think generally speaking, do cannabis plants produce alkaloids? The answer is no, they're not really specialized to produce alkaloids. Are you going to find evidence of maybe some strains that do produce minor quantities of alkaloids? Sure. And I'm not discounting that alkaloids I'm not saying that they're impossible or anything, but what I'm trying to get at is when you look at the biology of these plants, like we're talking about so much of their energy flowing down the terpene related pathways and phenolic related pathways. We see beautiful pigmentation and anthocyanin expression in plants because that kind of dovetails into polyketides. It's a little bit different, it's a little bit disjointed, and I am kind of reaching a little bit here, but my point is that flavonoids and purple pigments have a lot more to do with cannabinoids than alkaloids do. So like when we're looking at what do cannabis plants, generally speaking, what are they expressing? We're finding terpenes, we're finding phenolic compounds. I don't think we're finding too many alkaloids. I think there have been some that have been characterized, maybe a handful or so, but they may be present in the parts per billion, in the parent material. So volatile sulfur compounds, they are detectable to our nose in the parts per trillion. So even though it may be present in a small quantity, if that's indeed the case, that it's in roadkill skunk and contributes, if not defines, the aromatic profile, the odor profile, that it doesn't have to be in a super high concentration for it to be active on the nose. I think a lot of the compounds that are in some of the really fruity, like tropical fruity strains, if you get like mango terps or passion fruit terps or something like that, those could be volatile sulfur compounds that are only present in the parts per trillion. And parts per trillion, like it's 0.000000001%, I think, or plus or minus a couple zeros, whatever it is. But it's a small, small number. If you looked at a lab report, you'd see 30% cannabinoids. It's 30 point. You've got the dot, like you've got numbers in front of the dot, but in the parts per trillion, you have 0.00. You have a lot of zeros that lead up to it. So my point is, when we feed sulfur in the parts per million, you have to understand that if sulfur was a bottleneck for the expression of volatile sulfur compounds, you could feed 1 ppm of sulfur to a plant, and a 1 ppm increase would result in a massive increase in volatile sulfur compounds that are otherwise measured in the parts per trillion. Those are orders of magnitude difference. We're talking about saturating the substrate pool by thousands of times over what the plants effectively need to make the volatile sulfur compounds. So to me, that suggests a little bit more complex of a biochemical territory or a landscape that the plants have to navigate simply, beyond, is there enough sulfate present? Do I have enough Epsom salt in the mix to get my roadkill skunk terps? Maybe it is the alkaloids that are coming through, or maybe it is some combination of alkaloids and volatile sulfur compounds that are kind of coming together and creating a unique fingerprint, an olfactory fingerprint that just stimulates the brain in the right way. But the one thing that I do want to be careful of, particularly with air, anything that smells, anything that gives you like a distinct aroma that you perceive, that's going to be limited to between, I want to say between like 10 and 20 carbon atoms long. In other words, once you start surpassing a particular molecular weight above 20 carbons, 25 carbons, you don't have any olfactory properties at all. It stops smelling altogether. And if you drop too low with very low molecular weight fatty acids, for example, they tend to not have much of a smell, but the short chain fatty acids, those do have a little bit of an aroma. But everything that you can smell falls within a very narrow range of how many carbon atoms are actually on that molecule. And when you exceed a certain amount, it doesn't smell anymore. And when you go below a certain amount, it doesn't smell anymore either. So there's a lot of potential for molecules that may smell identical to other ones that are actually not that molecule at all, or it's like a different molecule that gives rise to that familiar scent, if that kind of makes sense.
Thank you for entertaining my madness and my crazy hypothesis.
Always.
I appreciate it. Here's Ken said that there might be some correlation here. Not quite sure, just crazy speculation.
Yeah, and if it comes down to volatile sulfur compounds, those are produced by hops. That's why hops has a similar aroma to cannabis, some strains of hops. And then, we did a really nice episode with Dr. Thomas YellowHammer from Oregon State University, Northwestern professor of fermentation science, if I recall correctly. And we spoke a little bit about how hops plants produce these volatile sulfur compounds that, the biosynthetic processes are really interesting too, because they're first, the substrate pool is built up, these parent molecules that are very stable and have thiol groups build up, and the concentration of those builds up to maybe a ppm or a parts per billion range. And then there's a process by which the substrate is acted on and it's released, and it produces this volatile sulfur compound that is measurable in the parts per trillion, but it is extremely potent and very, very strong. They're also very complex aromatics as well. They don't just smell like one thing. You couldn't just pin your finger to like, oh, this smells like that distinctly. You'd be reminded of multiple things. It's not like, if you've ever smelled myrcene by itself or linalool by itself, like linalool reminds me of at least half a dozen plants in ways that I just never realized these plants would ever be connected, except for smelling this one distinct terpene that they all happen to produce. But I can't trace it in any one of those individually. You know what I mean? Like lavender and black pepper, for example. Linalool for me is like this, man, it does both. It's sharp, peppery, it's spicy, but at the same time, it's like deeply floral. It has this like really interesting topical anesthetic quality to it as well. You put it in your mouth and boom, just numb right away. So, but with the volatile sulfur compounds that are produced by cannabis plants, there's some parent substrates that are formed and then they're oxidized and the volatile sulfur compounds are released. And depending on the, I guess, how far developed the trichome heads are, you may have different signature aromas coming out because these things are so complex. It could smell like anything from fresh cut tropical fruit, think mangoes, passion fruit, lychee, all the way to you put a skunk in a bag and beat the crap out of it like a piñata. It's going to smell like onion, funk, garlic, sweat, armpit. Like if you've ever had beer like that, that's what it is. That's the volatile sulfur terps that oxidize. And actually to that point, that's, I believe, if I recall correctly from the conversation with Dr. YellowHammer, the reason that Heineken uses green bottles is because that specific wavelength of light that it filters out prevents the degradation of those compounds, so you get a fresh hoppy taste out of it rather than like a stale kind of sweated onion flavor profile out of it. So yeah, to wrap this point up real quick, I'll say these volatile sulfur compounds, even though they're accumulated in the parts per trillion, they have remarkably complex olfactory properties. They can smell like anything from fresh cut tropical fruits all the way to dirty socks and sweaty onions and armpits and everything in between.
Sorry to sidetrack the conversation so much with that. I know we had a couple other concepts we wanted to get through. We're about 50 minutes in. I know we had talked about CO2 on the call the other day. We were going there a little bit about genetics and epigenetics and nutrition. I know we were kind of flirting with that idea, but anything else that you'd like to say about the sugar phosphate concepts?
Yeah, I mean, it's something that we've been working with pretty extensively over the past year, specifically with Peak Bloom, our equivalent of PK booster. And it's been a real pleasure just to watch the plants respond to sugar phosphates. And it's something that I, up until about a year and a half, 2 years ago, like, I could have told you that plants produce sugars through photosynthesis, and one of those sugars is G3P, but it never really occurred to me to try to figure out how I can replicate an environment that allows me to basically create and feed G3P and E4P and X5P and IP6 to the plants. These are all like the 3, 4, 5, 6 carbon sugar. These specific nodes that sit along our little phosphorus spine that we're watching branch out into all these metabolic pathways that just scream. I mean, cannabis plants just go wide open to the extent that they can on these pathways. These aren't just like little trickles of energy down here. Like the more energy we put on the plants, the more we watch these pathways get expressed, almost to the point of so much energy that it causes stress. But the way that the plants deal with the stress is to produce terpenes that help mitigate the stress. It's like so much energy. It's hard to describe. It's like being in sixth gear, going 150 miles an hour, flooring it, and then getting wheel spin and like trying to maintain traction. Like the plants are doing, crazy stuff like that. So, I mean, yeah, injecting sugar phosphates directly into their metabolic pathways is something something that we've been actively doing over the past year. It's been tremendously successful for us. And I do want to give a shout out as well to the agave and the artichoke that we're fermenting too. It's like we have this, we call it a sugar phosphate scaffold, if you will. We've got the usual suspects that we talked about with our little sugar phosphate spine, but we've also got some more complex sugars that are coming in through the fermentation of artichoke, which has some really cool fructo-oligosaccharides. And then we've got a really nice organic agave that we're working with. And some of you may actually have received a batch sometime last year. There was a batch of Peak Bloom that over fermented, and we ended up producing something that had like 14% alcohol inside of it. So if you got a bottle of Peak Bloom and it was swollen and it smelled like tequila, that's why the sugar phosphate fermentation chemistry side of the equation is just wild. It really is. It's taken us about a year to fully like stabilize it and dial it in, but yeah, everything's rocking now. So yeah.
Man, I'm bummed I didn't get one of those bottles.
Dude, it smelled so good. And I will say, like, we've had
there was a noticeable increase in the pleasurable aroma of Gen 3 for sure, but I didn't get one that was like near explosive, like really going with the deep notes. That's pretty cool.
Yeah, and we've had a couple of batches of product over ferment. Like Solar Rain was the only other one that fermented. It's not very often that it happens, but sometimes the fermentation runs away and it ends up producing a very high alcohol content in the final solution. So by the time you dilute it, it's not harmful. The plants actually like it. The plants actually do really well if you spray a little bit of alcohol on them. So there's maybe a mild disinfectant property on the phyllosphere. You spray the leaves with a little bit of alcohol and it can help kind of sanitize things, whatever. I think that there's something to be said about ethanol being produced and the plants just kind of getting drunk off it. Like some of the best results that we've ever seen with Solar Rain specifically came from those batches that over fermented, and we had an actual like kombucha culture. It was a SCOBY that was floating on top. We had alcohol distinct bubbles. Like it was because there's a lot of matcha that goes into Solar Rain, so it does truly ferment like a kombucha. But we had one batch in particular that got really alcoholic, and I was just like, let's spray it on the plants, because ethanol itself is not it's not harmful on contact for the plants. So I had no concerns about it being harmful. I wasn't like, let's spray it and see what happens. I was like, we've got a batch that fermented and produced some alcohol. Let's see if there's lots of beneficial effects, because there theoretically should be. And yeah, the Solar Rain and Peak Bloom go great together. So we had a batch of, alcoholic Solar Rain and a batch of alcoholic Peak Bloom, and the plants just freaking loved it, man. So I always say plants like to get drunk on, just fermented sugars now.
That's
like to
hilarious.
drink. Yeah.
I haven't seen many questions from the chat today. I imagine most people are absorbing this new stuff like I am. Josh seems super hip to most of it, but if anyone's got any questions or wants Nick to expand on any of these ideas, feel free to throw them in the chat. Get some more talking points going. Which I'm surprised. Usually there's tons of questions in the chat. This is definitely like some.
Yeah. And just to kind of recap the point about skunk terpenes and all that stuff, I mean, part of the elusive nature of it is that those compounds, which are typically associated with those aromatics, those are not produced in super high concentrations. So you actually need people, I forget who the researchers were back in 2019 that did that really nice study on volatile sulfur compounds. The way that they put that study together was genius. It was very intelligent, and you need that kind of creative level of thinking if you're going to quantify and determine something to the nearest part per trillion. Like you got to get smart with how you're going to be running your analytics. So it's been a while since I've looked at that paper. I wish I could speak more specifically to how they had set it up, but I'm sure everyone's familiar with that study. So yeah.
Here's a couple questions that popped up, Nick. What are if any changes to the resin bloom for Gen 3?
Yeah, I mean, resin blooms all.
You can talk about it if it's not like too sensitive of like inside stuff or whatever.
Yeah, no. I always try to give as much information to people because the proprietary stuff of what we're doing has a lot to do with process oriented. We're manufacturing the stuff from scratch. Like I'll bring in whole birch leaves, we bring in whole jasmine flowers. Like anything that's inside of the plants isn't really like a proprietary thing. So I love talking about what's inside of the plants. I love talking about what's inside of the fertilizer ingredients themselves, because we're making the stuff from scratch ourselves. It's not like I have to, be careful about anything, because we're not talking about process, specific stuff. So resin bloom, gosh, it's always been one of the best performing products, and it's experienced probably the least amount of change between when we first started and where it's at now. But what I will say specifically that kind of leads to a discussion, we were talking earlier about IP6 or inositol phosphate. It's that 6-carbon C6 that plants are working with, and it is present inside of cannabis trichome heads. But the thing that happened with Gen 3 resin bloom was more along the lines of, can we use these sugar phosphates to specifically plug into pathways that we see being expressed in abundance in cannabis trichome heads? If we're looking in, and we see a lot of enzymes associated with pentose phosphate metabolism in plants, I wonder if we can somehow, work with sugar phosphates to deliver those. So the NPK profile of resin bloom more or less stayed the same. It didn't really change. But the specific sugar phosphate load, as it informs trichome head biochemistry specifically, is what the major focus was. And what I'll say specifically to that point is there's a reason that birch leaf is now on the Powered by Plant stamp. It's a little bit of a subtle reason, but it's not like you have to dig very hard to try to find out exactly what I'm talking about. In fact, it's somewhat apparent by the use of what birch is already kind of used for anyways. And that has some similarity. I will say it's not a direct translation into cannabis and cannabinoid biosynthesis and trichome head chemistry. But there's a bridge there that involves a 5-carbon sugar. So yeah, I mean, resin bloom has always been my favorite.
I believe you said that was your flagship product, the first conversation we had. I remember asking.
In terms of customer feedback, I will say resin bloom is one of those products I can give somebody that is just about to use our products for the first time and they only have 2 weeks left on their flower. So like, how big of a difference could it possibly make, right? It's not going to be that noticeable. We'll just add a little resin bloom, like 2 to 4 mils per gallon, and the plants all maybe see a little bit of change. And it's like, no, people come back and they're like, there was a noticeable change. They have, experience with some genetics they've been growing for the past 15, 20 years, and they just use resin bloom, and they come back and they're like, this is interesting because I only had 2 to 3 weeks left on the plant's life, and like it got steered in this totally different direction. And part of it is the carbon as a substrate, and then the other part of it is that it's chemically reduced carbon, so those electrons are rolling downhill to the favor of plant metabolism. The energy is being released within the plants. It does not require energy expenditure to access these forms of nutrition. It's actually kind of the opposite. As soon as the plants get them, they get an increase in the energy states that they exist in. So their energy landscape environment changes fundamentally. And if you couple that directly with compounds that are not only produced in glandular trichome heads, but also accumulated in pretty significant concentrations. Again, some of these compounds I was mentioning can actually match or exceed the concentration of terpenes in total that are in the actual trichome head. So you have to think these things are important in the overall expression of what's going on inside of these trichome heads. Very complex environments,
Someone made a comment here. Was I wrong? And you said Solar Rain was the flagship? I thought you might have said that in later conversations, but I thought our first you said it was resin bloom. I could be wrong.
So that's why I clarified and said that.
I thought it changed over time. Like a year after us talking, I thought it was a Solar Bloom, but I could be wrong.
Well, so that's why I added that caveat of, by way of customer feedback, Solar Rain, resin bloom is the customer favorite, but the actual flagship product, if you were to ask me, I would say it's Solar Rain, because Solar Rain really represents this idea that we can take carbon as a macronutrient very seriously, and we can do things like wrap this complex language around what we're doing, chemically reduced carbon, feeding directly into these substrates along these specific nodes of sugar phosphate spines and all this wonderful stuff. We've been talking about this stuff for years. This is not like I'm trying to dump a bunch of information in front of everybody and say good luck trying to figure this out. I mean, we had dozens of hours of interview time where we're talking about these very nuanced concepts. We did very long episodes on redox chemistry and the electron transport chain and all this
I feel
stuff.
like the conceptual continuity of them as well, like they kind of always led into the next concept. And then the foundation of that led into the next concept. I feel like anyone who's really been rocking with your broadcast, like definitely have gone down the path, like.
Yeah, and along those lines, the path that we've been coming to ultimately has been to talk about sugar phosphates, because we've talked about all this like electron transport stuff and redox chemistry as it pertains and leads up to photosynthesis, but we've never talked about what is the product of photosynthesis. We talked about everything that leads up to it. We talked in great detail about PS2, PS1. We talked about the water-water cycle in PS1. We talked about the oxygen evolution we spent like an hour and a half talking about just the oxygen evolution complex in photosystem 2 and how it depressurizes water molecules in the most intense electric field generated by any organism on Earth. We spent a very long time talking about that, but we forgot to mention, turns out plants produce sugar phosphates through photosynthesis. And that's what we're talking about now. So in a lot of ways, this is an extension and continuity of the things that we've already discussed to a pretty heroic extent, I feel like. And I appreciate everybody that has stuck around for the past few years and listened to the evolution of these discussions, because you've watched me learn along the way, and I hope that you guys have also learned along the way too.
How you doing, Josh? You still there, brother?
Oh yeah, absolutely.
Right on.
Yeah, just thinking a little bit about this product, solar rain you were just talking about. And so is that meant specifically as like a foliar?
Yeah. Yeah. And if you'll entertain an idea that I have about solar rain, I do want to answer your question about solar rain, but in case I forget, just remind me that there's something that I wanted to, an idea that I wanted you guys to entertain about it. But yeah, I'm sorry, what was the specific thing about solar rain?
Well, no, I just was looking at it's, primarily a magnesium product.
And that's your only one that's foliar only. Like that one you don't recommend as a root drench, right, Nick?
Yeah, for Gen 3 solar rain, you can water it in.
Oh, cool.
At like 1 to 2.5 mils per gallon seems to be pretty good. And then the complementary product to that, root anchor, which initially was intended for soil drench only without being applied as a foliar spray, you can now do root anchor as a foliar spray as well. So the two have kind of like bled into each other's function.
More utility. That's so cool. More versatility. So cool.
Yeah. So solar rain is like a really interesting idea wrapped up into a product, because the idea was basically at large, can we feed carbon to plants and have it behave like a macronutrient? If I put some calcium onto the plants or some magnesium and phosphorus, I can watch the plants absorb that. But I wonder if I could put a little bit of carbon on the plants and watch them absorb it too. And over the course of about 10 years, it ended up developing into, what it is now with Gen 3 solar rain. But along the way, it picked up these really, really cool features. And it's not so much that the magnesium, so the format of the magnesium, I think, is very important, because the specific chelates and complexes that we're making for, those are absorbed across foliar surfaces with a remarkably high degree of efficiency. And primarily those would be the organic acid chelates and complexes, the acetate form, magnesium acetate, magnesium citrate. The environment that the chelates and complexes are surrounded in is an environment that's very conducive for, there's an enzyme called magnesium chelatase, and its job is to insert magnesium into the porphyrin ring of chlorophyll. So it kind of gives that stamp of magnesium, and then you have this final molecule chlorophyll that's produced. The pigment is finally produced. So the environment that the chlorophyll magnesium chelatase operates in is one that is maximized in terms of chemical gradients, most conducive for the formation of that chelate downstream. So there's a bunch of cool stuff. I mean, aside from the actual organic acid complexes and chelates that are being made that are on the label, because these are full spectrum, derived from plants, the profile of the chelates and complexes is actually a lot more than just what's listed on the label. Like we're using orange peels, for example, to ferment. We're fermenting orange peels and then extracting the organic acid content out of the orange peels and using that to make a true calcium citrate fraction that we're maxing out the solubility limits of. It's not, supremely soluble in water, but my point is we're taking the organic acids out of orange peels and maxing out one specific fraction of solubility calcium citrate. There's also pectic acid residues. There's derivatives of organic acids like hydroxycitric acids maybe that are also found there. Probably 10 to 15 different organic acids per plant that contribute to this whole chelation and complexing chemistry thing that we have going on. And all of it is meant to power the processes that lead, downstream to photosynthesis. I will say one of the really cool things that we've been doing with Solar Rain that I haven't seen other people do is that we're making metalloanthocyanin complexes. So we're extracting a very specific kind of anthocyanin, and we're using that to complex it with magnesium because the anthocyanins themselves, they have hydroxyl groups that are reactive and they can participate in things like metal chelation, much like you would find with organic acids. It's actually the exact same fundamental mechanism that we see in organic acids occurring. So the formation of a metalloanthocyanin complex, specifically with magnesium, being delivered as a foliar spray is something that has profound benefits over something like an Epsom salt, a magnesium sulfate kind of compound. So, and that's maybe one of the layers that we can get into, as well as the use of some of these plant extracts and some of the compounds as chelating and complexing agents, because they have a variety of benefits beyond just increasing the solubility of compounds or beyond increasing the stability of the electrons and preventing them from lattice and forming insoluble salts and all this bad stuff we don't want to see happen. These plant extracts actually have remarkable benefits aside from that and outside of just that context. But before I get into that, I figured I'd let you, I hope I answered the question appropriately about Solar Rain there.
Yeah, no, absolutely. It's a pretty interesting concept for especially a foliar. I'm a huge fan of foliars and I've used them extensively. You can't really magnesium plays such an important role, obviously, in stabilizing chlorophyll, but more importantly, in line with our discussion overall, it really supports electron transport efficiency, and the effect essentially becomes a much smoother electron flow and reduced ROS formation under high light. So if you're out there and you're listening and you're running a high ppfd, this product should work well for you, because it can help support some metabolic flexibility and reduce the transient redox imbalances that can form. Because if, like, essentially your plants need the ability to reduce to for terpene and cannabinoid synthesis, you have to have these reducing equivalents like NADPH, and essentially using this and magnesium helps to produce that redox sink that allows the formation of these compounds. Otherwise, the system just kind of backs itself up. If you get a redox imbalance and you get NADPH accumulating, the Calvin cycle is going to slow. You're going to see G3P production go down, which leads to a reduction in carbon flux. And so redox balance essentially determines whether the sugar phosphate production can continue. And so this is kind of a foundational element to making all of this work.
Yeah, and one of the other things that I'd like to toss into the fold too is this idea that when we're looking at the function of anthocyanins, that they can absorb light energy that can be actually calculated, right? We've got Planck's equation that we can use to figure out what is the specific energy that's carried by each wavelength of light. And the closer we get to ultraviolet, the more we realize like plants have to have some kind of protective mechanisms in place. Otherwise, that can quickly lead to production of reactive oxygen species, particularly if we have overexcitation of the electron transport chain, or if we have, the reactive oxygen species part of this, comes from the fact that there's just oxygen is a constituent of the atmosphere. Like, you're not going to get away from it here on Earth. And plants, of course, being terrestrial, they have to deal with, let me see if I can move this a little bit so it's not as, they have to deal with the oxygen that's in the air. So part of this is they have to balance the redox state of oxygen that's present inside of their own tissues. And if they allow for oxygen to just run away and grab all the electrons, it's going to quickly rob the electron transport chain. It's going to result in irreversible damage and things like that, which occur. The likelihood of that damage occurring with frequencies, like you're more likely to have that damage occur the closer you approach ultraviolet wavelengths. UV light tends to be a higher intensity, it carries more energy, and as a direct result of carrying more energy, it's more likely to damage the photosynthetic machinery within the plants. I'm going to see if I can get around this way. The idea that I'll toss out is that anthocyanins, when applied as foliar sprays, could be viewed as a new kind of a class of biostimulants for plants, because typically when we're talking about biostimulants, we're looking at things like what role do they play in plant signaling. This would be something like a kelp extract, some kind of very specific sugar extract that may be more complex sugar, like a fructooligosaccharide from artichoke, as opposed to just a sugar as an actual building block. We have to, I've kind of lost my train of thought there. What were we talking about? I'm trying to move around here.
You were mentioning anthocyanins as a potential foliar.
Oh, yeah. So anthocyanins will absorb specific frequencies of light that otherwise cause damage to photosynthetic machinery. And the idea here is that we've got classes of biostimulants that function as signaling compounds. They pass a message along, or they somehow stimulate some downstream thing to happen. The second class of biostimulant might be that there's a carbon substrate that's actually provided. There's some carbon coming in, and the plants will use that carbon to actually make a compound. The third class of biostimulant, this one that I'm trying to talk about now and to try to introduce, is the idea that anthocyanins can behave like optical brakes for plants. If you have high light intensity and that light intensity is driving the formation of reactive oxygen species, then maybe foliar sprays of anthocyanins can absorb those light frequencies that would otherwise cause damage to the photosynthetic machinery. This has nothing to do with the way that anthocyanins interface in plant biology. If we were looking at anthocyanins and their natural context, how they're natively embedded in plant biology, we would be looking at a similar function here. But I'm talking about removing the anthocyanin entirely from the plant and having just the externally applied anthocyanin be what causes the reduction in reactive oxygen species formation. And again, it's the specific interaction between the chromophore, the flavillum cation that sits at the center of that anthocyanin, and that specific frequency of light, somewhere around ultraviolet, maybe extending a little bit closer towards green light spectrum. Those wavelengths of light cause damage, and they can effectively be absorbed by anthocyanins. That absorption does not require plant biology. It can actually happen outside of the context of plant biology. So just a little bit of, I guess, food for thought. I don't know. What do you think about that?
That's really interesting. That's really interesting concepts. Especially, I've never really thought about anthocyanins as a biostimulant.
Yeah, as an optical brake more than anything, because they basically take high energy wavelengths of light and step them down so they get released as harmless phonons, vibration, heat, basically, and this is a really profound thing that plants have engineered ways to do. But the interesting part from my perspective is that oftentimes when we look at the function that some of these protective compounds play within plants, we almost have to suspend the compound within the native plant biology to see that functioning properly. But anthocyanins are this really interesting class of compounds whose electrons interact directly with photons from the sun with no biology interface required whatsoever. So the anthocyanins that are produced by plants, like in the berries, for example, and the fruits and flowers. We can extract those and then do foliar sprays of those on cannabis plants in the same light absorbing properties and benefits that they have to offset or behave like an optical break within specific band gaps that are otherwise very intense for plants. We can see that happen, and it's pretty remarkable, because I'm not sure, to what extent you're familiar with anthocyanins, but like in terms of optical density, they're remarkable. You don't need much anthocyanin to get huge absorption, and they're recyclable within plants in a natural kind of setting. And if you created the right environment for them, with flavonoids and other molecules, then they might be complexed or co-pigmented with natively in plant biology. You can extend their function so they can repetitively absorb frequencies of light and release them as harmless vibrations without actually breaking down. And this is a stabilized in solution is what I'm talking about. This is outside of plant biology. And it has a lot to do with what we're doing in solar rain and how we're making the metalloanthocyanin complex. I'll give you a hint as to what I'm talking about. The pH of that product in concentrate is extremely low. And then even when you dilute it at 10 to 20 milliliters per gallon, the pH of the spray can be around 4.3 to 4.5. And that's all driven by organic acids, free organic acids. So there's a powerful reducing environment that is created. And the reducing environments are highly beneficial to the prolonged and extended function of anthocyanins. It's very specific kinds of anthocyanins too, I will say. The aronia berry stamp is on there for a good reason.
Yeah, this is a really cool topic that for me, it's something I've not really thought about or explored. It's kind of got my gears turning. I mean, especially as the anthocyanins, like the role they play just in plants typically. Really, really interesting take.
Yeah, I mean, they're redox active molecules too, right? When we're talking about anthocyanins, there's this core molecule that has some level of redox activity. Depending on which one of the 6 major classes we're talking about, we're dealing with slightly different redox properties. But if you remove the sugars that it's attached with, on the top and bottom, glycosylation. So basically what I'm talking about is like for the audience here, this is like, imagine I gave you a sandwich. You got your bread and you've got your meat in the sandwich, right? The meat is the part of the anthocyanin that actually absorbs the light and interacts directly with the light. And it's also the thing that has the electrochemical properties, the redox active properties. The bread or the sugar are responsible for this. They modify things like what happens, where does it go, where does it get accumulated, where does it get transported. So that's the sort of way that plants will produce compounds that are not soluble in water, put sugar, bread on them, and then basically allow them to be transported in water. Sorry, Josh, what were you going to say?
Nothing. I forgot to mute my mic. Sorry. I think I was just making noise in the background.
What do you got going on, Travis? You want to say something?
No, I'm on mute. Yeah, a bunch of questions are coming in. I was waiting. I didn't want to steer the conversation, or it seemed like you guys were riffing, but there's a bunch of questions that have been coming in if you wanted to get anything, unless you wanted to steer the conversation in a specific direction.
No, we could do questions. Love to do Q&A.
Cool. Here's one from Toad. I think he's talking about the solar rain specifically, but asking if it can be watered in along with foliar applications.
Yeah, it's a good way to get a couple extra ppms of magnesium to the plants. Somewhere around 1 to 2.5 mils per gallon seems to be the sweet spot. The magnesium chelate and complex package is different than the one that we're making for CalMag Fuel. So CalMag Fuel has 1% magnesium inside of it, and Solar Rain also has 1% magnesium inside of it. But the two are fundamentally different in terms of their effects. And even in the soil versus as a foliar spray, the reason that past generations of Solar Rain have not been recommended for use as a soil drench is specifically because that chelate and complex is engineered for leaf surfaces. It has a different effect in soil biology. But now Gen 3 can be watered in at the rate of about 1 to 2.5 mils per gallon seems to be the sweet spot. We've had some people go beyond that, but I think that if you're running our products, like there's magnesium inside of, the base nutrients, there's magnesium inside of CalMag, obviously, a little bit of magnesium inside of Peak Bloom now. And to a point that Josh was mentioning earlier about magnesium and cell energetics, the relationship between mag and phos is super important. So, we've got magnesium coming in from multiple different products, but the forms of magnesium in each one of them is different. There's no two that are identical or even similar to each other. But Solar Rain is definitely the flagship. That's the specialist sauce. In fact, there are some derivatives of the Solar Rain chelate that actually got pulled out and converted into the CalMag chelate as well. So it's like everything sits downstream from Solar Rain. It's like the progenitor of it all.
I will say from experience, I don't think it's like lethally toxic, because I remembered I think it was my second or third one, I grabbed the Solar Rain container by accident, and I did a liberal dose of it as a roots drench, and I didn't realize it till after, and it seemed all right. They were fine.
Yeah, I mean, it's not.
It didn't kill anything, not even close to that. You couldn't even tell. Grumpy.
No, and it's more of a specific nuance of how those organic acids are metabolized in the soil and what the products of that metabolism lead to downstream. In foliar surfaces, it's a fundamentally different environment, so we can drive the conversion of these compounds that we're putting in the solution much more predictably, across the foliar surfaces. But in the soil, like I said, for Gen 3 Solar Rain, it's not a problem to water it in. You should be okay. We haven't seen any negative effects whatsoever. The magnesium content is still just as high at 1%. But the other thing that I want to point out too about the products, Solar Rain's probably a good one to talk about because it's 1% magnesium. So, I oftentimes hear people say like, oh, it's 1% magnesium, it must be 99% water. And I'm like, dude, Solar Rain weighs 9.5 pounds per gallon. Like water weighs 8.3 and Solar Rain weighs 9.5. Look at any label of any foliar spray that's out there, any kind of fertilizer that's out there, and tell me what comes back as 1%, only 1% magnesium and weighing 9.5 pounds per gallon. The rest of the stuff that's in there is carbon. That's why it's 15% carbon, 1% magnesium, and it weighs 9.5 pounds per gallon. Like when you pick up Solar Rain, you're like, whoa, this weighs a lot more than plain water does. So no, it's not plain water. Yes, there's a ton of carbon inside of it. It's guaranteed at 15% carbon, and all of that is chemically reduced carbon. So those electrons are just waiting to roll downhill. As soon as the plants touch it, it starts to interface via electric fields and all this wonderful tingly stuff that happens. So yeah,
I feel like you get super stoked when you're able to have a heavier gallon too. Like if you can increase 60 70 grams on a gallon, like the couple times we've talked, you're stoked about it. It's pretty cool.
Yeah, because it's all carbon and
yeah,
and we were also talking about like the greatest ROI that you'll ever get in your garden is figuring out how to feed more carbon. You could feed twice as much of your, amino acids, or you could feed twice as much of your CalMag, or you could make the fans twice as powerful. You can turn on your lights twice as much, double the intensity, of the lights. Those won't double the yield nearly as simply, cleanly, and straightforwardly as doubling the carbon on the plants. If you figure out how to increase the carbon, you absolutely will increase your ROI. It's just 100%, no doubt.
On a side note, what'd you say, Nick?
I think Josh wanted to chime in with something.
Yeah. Well, I was just going to ask you, were you seeing that the application rates for Solar Rain were 1 mil per gallon?
Yeah, for a soil drench, 1 to 2.5 milliliters per gallon. Yeah.
And what's the rate for foliar feed?
10 to 20 mils per gallon.
I mean, that's still like really, compared to a lot of other products, that's, 1 to 2 mils per gallon for a soil drench. That's nothing. And that's just, most other products you're putting in ounces, like you're putting in 60, 90, 120 mils. So that's remarkable. Especially thinking just like for larger grows and commercial, cultivators. Like, you go through a lot of products because you're having to put, you're mixing up larger batches, but you're putting in, 50-pound bags of this or that or whatever. But that's impressive. That's a concentrated formula.
Yeah, and it works so well, in my opinion, because of that carbon to macronutrient ratio. We're putting 1 ppm of carbon, 1 ppm of magnesium into the plants for every 15 ppm of carbon that gets worked in. So it does something to the energy state of a plant because it's chemically reduced carbon. So like I was mentioning earlier, those electrons are just waiting to be released. They come in a variety of different forms, like organic acids that plug directly into mitochondrial function. Magnesium citrate is one of the key constituents of solar rain. And for those of you who are familiar with the mitochondria, they have a citric acid cycle that turns out if you supply exogenous citric acid to, you can increase the throughput of that cycle. And even for photosystem too, the oxygen evolution complex, metabolism of citrate can actually help increase on a sort of a quantum physics basis. You can increase the throughput of your electron transfer because it's almost like there's this little lightning strike event that has to occur in order for the electron transport chain to happen successfully. You need a sink for the reduction power to flow towards. So this is the sort of hardwire between PS1 and PS2 and how the electrons flow. But it turns out if you supply little sinks that reduction power can be stored in, some of the research that's been coming out, especially over the past 5 years, has indicated that there are effects with things like wave function collapse of the exciton and the dedicated transport chain being activated, because in the reaction centers anyways, this may be a little bit too much for people. So let me, feel free to interrupt me if this is a little bit too far, but the reaction centers of the chloroplasts or where the actual true charge separation happens. It's not like every single photon of light that comes in excites an electron, and that electron then participates in true charge separation. When you have the light harvesting complexes, the electrons stay in the chlorophyll pigments. So this light energy comes in, and it starts to vibrate the electrons. They go up, but they fall back down in the same chlorophyll pigment. That's out in the light harvesting complexes because the distance between the chlorophyll pigments is so great that the electrons actually don't have overlap sufficient enough to drive one electron up and allow it to hop across over to another pigment molecule. In the reaction centers of PS2, the ones that are specifically tuned to 680 nanometers, those ones, the chlorophyll pigments, the special pair is placed a lot more closely together. And they are so close together that the magnesium ions share an overlap, a little pi overlap inside of sp2 or whatever. I forget the electron shell specifically, but this overlap allows for light energy to come in, and there's an excited electron whose wave function is actually shared by both chlorophyll pigments. And if one of them has a downstream receptor, that wave function collapses, and you get true charge separation occurring in the reaction centers because the electron transport chain has a sink. This is kind of what you were talking about earlier, Josh, with back pressure basically building up in the electron transport chain at the site of NADPH as the acceptor of that reduction power that flows, between PS2 and PS1. So that was kind of like the far deep off quantum physics rabbit hole. We don't have to go super far down that, but yeah, citric acid, very important. Feeding organic acids that have reduced forms of carbon that have the electron potential ready to rock and roll can go a very long way for figuring out how to activate plants expression, how to feed the terpene and the cannabinoid expression, because those require a lot of energy ultimately.
I'm going to give a tactical pause in between each break now, Josh, in case you have something to say. I feel like I'm always quick to just start running my mouth. So if you notice an awkward pause, that's always for you if you want.
Oh, sorry. All right. I didn't know if you had another question from somebody in the in the audience, or
There's a whole bunch of questions for sure, but I want to try to take a pause each time instead of just getting right back to it in case you had something you want to say. I feel like this question from Sapo is a great tie-in to kind of what we were talking about the other day as well. How efficient is carbon absorption through foliar application compared to CO2 injection in the air, or are they even comparable? And I would say, I guess, from the substrate as well. And yeah, we've talked about this a couple times, how much more energy it takes. You know, it's not like the carbon just separates itself from the O2 in the air and kind of just comes in the leaf. It's like, it's a lot of hard business going on there from what you've explained it. And it seems like if you could bypass that, it's much more efficient. But
yeah, I mean, this is a very good question, because it is a little bit of a rabbit hole of a topic. Now, CO2, and if you want to keep that question up, that actually kind of helped me stay on track too, because I have a feeling I'm going to go off topic a little bit here for this one. CO2 injection in the air, like, it's not the O2 in the CO2 molecule that's important for the plants. It's actually the carbon that they're trying to get out of the air. So they have this process by which they take the energy of the sun and they use that to convert CO2 molecules out of the air into sugar phosphate form. That requires NADPH, which is generated through the electron transport chain. It requires ATP that relies on the function of the oxygen evolution complex, splitting molecules of water apart, generating electrons that feed into the electron transport chain, and then also generating protons that feed downstream into the function of releasing CO2 to Rubisco, and then also participating in ATP biosynthesis, which, just to touch on one point real quick that Josh brought up, magnesium and cell energetics. Magnesium, also the highest concentration of free magnesium that you're going to find in a plant, revolves around what's called the adenylate kinase equilibrium. Adenylate kinase equilibrium. Adenylates are things like AMP, adenosine monophosphate, adenosine diphosphate, ADP, and then the famous one, the most famous one that everyone's familiar with, which is ATP, adenosine triphosphate. So you've got this pool of molecules that exist in a mono, di, and triphosphate form of each other. Magnesium is actually balancing out the ratios and the roles between all of them. You've got this kinase enzyme that facilitates the rapid interconversion of ATP molecules with, ADP and AMP molecules. Even the product of ATP synthase itself is magnesium-ATP. There's no such thing as free ATP that is a product of ATP synthase. It's magnesium bound. So most of the ATP that's inside of plants, about 99.9% of ATP that's inside of plants, is going to be chemically bound to magnesium. So yeah, magnesium plays a really important role, not only in photosynthesis, in electron transport via chlorophyll, but also further downstream when ATP is being manufactured using the light energy, by the way, that magnesium also helped capture. Magnesium pools are responsible for keeping the ratios of AMP, ADP, and ATP optimized so that the product, which again contains magnesium, which is produced, fits into an equilibrium mechanism at play here. The amount of free magnesium versus the amount of bound magnesium creates this function, if you will, within the plants. And that's why adenylate kinase happens to be one of the most profound enzymes in nature. I think it's a diffusion-limited enzyme as well. But even further downstream from that, when we're looking at Rubisco and CO2, when we're talking about putting CO2 into the air, Rubisco is the enzyme that takes the CO2 out of the air and introduces it into a phosphate sugar. As it turns out, the catalytic center of Rubisco relies on magnesium as well. So magnesium is doing this thing in chlorophyll where it's helping convert light energy into chemical energy. It's doing this thing with sugar phosphates where it actually helps inform the creation of ATP. And then also for, not for sugar phosphates, for adenylates that are very important for plants and for sugar phosphates. And then finally, on the photosynthesis side, on the actual Calvin cycle side, we see magnesium pop up yet again. And we find it participating directly in the carboxylation activity. So whenever Rubisco takes CO2 out of the air, magnesium is involved in a specific chemical reaction that allows the CO2 to get transferred effectively. So it does become very important for magnesium to be present all the way around. Now, how efficient is carbon absorption through foliar application? It depends on the specific form, because when we're talking about introducing CO2 to the plants, that entry point is in the Calvin cycle. The Calvin cycle is a very slow cycle because Rubisco takes about 3 to 10 seconds, somewhere in that range, to fire once. So you give your plants a molecule of CO2, and then for it to fit into the engine and fire and convert into a sugar may take anywhere from 3 to 10 seconds. It's kind of a slow process. If you could figure out a way to put the carbon in a form where the plants don't have to spend 3 to 10 seconds and require the power of the sun to convert it, that might actually yield some kind of benefit just in terms of time, because you've managed to take a shortcut. And then also maybe an increase in the energy state of a plant too, because rather than requiring energy to break the carbon away from the oxygen molecules, what if you already, and it's more about the landscape of electrons more than it is about the relationship between carbon and oxygen. The carbonyl group itself, that motif, is something that underpins all of biology, and there's some electron transfer properties, delocalized electron transfer properties, that have to get sort of considered. So it's not so much that oxygen is bad and carbon is good. They have a very profound relationship with each other. It's more about where are the electrons, like in the game of musical chairs, where have the electrons landed? They, generally speaking, will shift more towards oxygen. But if you can have specific kinds of molecules that donate electrons to biochemical processes, then the absorption of those carbon compounds becomes significantly more efficient than just CO2 in the air. The differences are that we're talking about injecting it at different points downstream. CO2 enters the Calvin cycle and it doesn't enter any other cycle. Well, I mean, yeah, I don't want to get too far into it. There's PEP carboxylase inside of trichome heads that's active, and it can help recycle CO2 in oxidizing environments. But that seems a little nuanced for the purpose of this question, I will say.
Yeah, I think one thing too that I would just mention from the grower's perspective in this question, specifically about getting more carbon, maintaining proper VPD during your grow is critical to keeping the stomata open so that they can intake the CO2 required for the carbon. I think a lot of growers miss that importance of VPD, especially during flowering. And if it's too low, your stomata become lazy essentially, and your transpiration is very weak. If your VPD is way too high, your stomata end up closing and your carbon intake really drops. So if you want to optimize carbon intake by the CO2 pathway, maintaining an optimal VPD during flower is critical, kind of between, so like 0.8 to 1.2 kilopascals.
Yeah, and I'm a fan of running on the dry side of the VPD curve. I feel like if you can pull more feed water through the plants, it ends up dumping more carbon into them. But I've also seen that there's compounds that you can feed the plants that will modify some of the pressure regimes that are present inside of them. Because the plants kind of sit at this halfway spot between the pressure regime in the soil, which we measure in megapascals, and as it approaches zero, we're reaching the saturation point, and then we move away from zero in the negative direction to signify a drier soil, basically. And vapor pressure deficit, on the flip side, kPa, kilopascals, it's interesting because as we approach zero, we approach 100% humidity, and we move away from zero in the positive direction, not in the negative direction, right? So we've got these two things going on in the soil like this, right? VPD, the higher the VPD is, the drier the air is, and the drier that the plants are. And in the soil, we move away from zero in the negative direction to talk about how dry the soil gets. And the plants are kind of like in between these two pressure regimes, where the delta between megapascals and kilopascals is such that the dynamics of whatever's going on in the soil will push their way through the plant and sort of override to some extent, not to a great extent, but to some extent they can override what is happening in the air. Now, ideally, you want that soil plant air continuum to be fully dialed in and VPD just being a sort of a constituent or a component of that, as a way to measure like what's the draw or the pull of that the air has. And if we can figure out what's the moisture retention capacity of the soil, coupled with some of the specifics of our equipment, like how much can my gear dehumidify, how many pints is it going to pull out in the sealed space, like we could start to build out some metrics of, what's ideal for the environment. Like, how's this going to work best? I have a certain moisture retention capacity in the soil. I know my plants want to take up that water and transpire it. And if I get the soil moisture and the VPD properly, that water flows right through the plants passively. They're just along for the ride. They don't even have to try at that point. They're just kind of there to help facilitate a natural process that's already occurring, and do so in a way where you're not maxing out your equipment. Because if you get too much water into the room and your pots are too big, your gear's just going to work too hard to try to pull out all that moisture from the soil. When in reality, smaller pot sizes with higher limitations for your equipment might actually be a better way to do it. That way you're not overloading your gear, and you're not just depositing a bunch of moisture into soil. It's going to take a while for your DUs to pull out. So, yeah.
Spacking it.
SPAC, that's right. Soil plant air continuum. It was a good little conversation.
Couple more questions if you wanted to entertain. Or did you have something more to say?
No, not specifically. I was just going to keep ranting like I always do. So please interrupt. By all means, do interrupt.
You're the best at that. Hell no, I'm not interrupting. I'm listening. Here's one about shelf stability. I will say that I have solar rain that I think is like damn near 4 years old. That is still pretty good. And I've had other ones that certain generations or versions seem to like change for sure, like whether it be the aroma or the texture.
Mm-hmm. Yeah, I mean, as far as shelf stability goes, they're all pretty much 12 to 18 months at minimum. If you keep them in the correct environment, like try not to let them freeze. Don't let them boil, don't let them, because they are products of fermentation chemistry. So if you expose them to environments that are above 80 degrees Fahrenheit, you might start growing some microbes inside of some of the products, which isn't necessarily a bad thing. Particularly with some of the products like Solar Rain, you absolutely will get a true kombucha SCOBY on top of that. Peak Bloom, you might get tequila, some kind of yeast that ferments and breaks. It's all natural and it's all good stuff. It's not going to hurt the plants. As far as some of the changes that happen on a batch by batch basis, we're starting with whole plants and we make stuff from scratch every single batch. Sometimes we work with different suppliers. Like we just started working with a farmer down in Oregon that's supplying us with artichoke now. This artichoke is freaking phenomenal. It's such a high quality artichoke that Peak Bloom, you can tell the difference with this artichoke. It smells so much better. So there's going to be little things like that just by nature of our business, these are very much so natural products that are sourced from whole plants that we're working with ourselves. We're not buying anyone's 55-gallon drum of kelp extract. We're getting the kelp ourselves and making the extract ourselves and not doing it in a chemical solvent based way. There's a lot of fermentation chemistry that goes into figuring out how we pull these molecules of interest out of these plants of interest and work with all of the stuff. So you will see some changes from batch to batch. A lot of it is just stuff that we can't necessarily control. But, as we move forward in time, those are the types of things that I'm personally really excited about stabilizing and dialing in. It's just the relationships we've got with farmers. Like there's one farmer here in Washington State that grows a lot of plants for us. And, we placed an order for some sage leaf, and this was like on a Thursday or Friday, and she got back to us and she said, I'm going to be harvesting some sage over the weekend. So on Monday or Tuesday after it's, fully dried out, we'll send it to you. And she sent it to us, and like that batch of sage, oh my God, it was so terpy, so aromatic, so fragrant. And those are the types of things that I really enjoy. This is why, I do what I do. It's a real honor and a real privilege to be able to work with plants to this extent. Like day in and day out, all I do is I touch, smell, eat, breathe 36 species of plants that we work with and turn into fertilizer. And just the aromatics, like all of this life that gets attracted to the manufacturing plant on days that we make like CalMag fuel, for example, we're grinding up a lot of orange peel. And so the whole plant smells super terpy. And there's birds that come, there's frogs, there's lizards. There's all kinds of like wildlife that's just checking it out. It's like, well, what's going on here? All this like very pungent, aromas and stuff. So it's like it's attracting life to what we're doing, and I feel really good about that. And supporting regenerative and sustainable agriculture directly. I can't tell you how happy I am that every year we spend, tens, if not hundreds of thousands of dollars. We give that amount of money directly to sustainable and regenerative agriculture. Like, we're definitely putting our money where our mouth is. We're supporting farmers directly by working with them directly, and they're growing plants for us. We're taking those plants and we're making fertilizer out of it. And it feels so good to be able to directly support sustainable and regenerative farmers directly through this mechanism. And it's something that I think sets us apart too. Not many other fertilizer companies are out there in the business of talking to farmers and getting fresh green biomass, very specific kinds of fresh green biomass too, in order to make fertilizer product. I remember the first time that the farmer that grows our aronia berries, I told him that I was going to use aronia berries to make a fertilizer. He looked at me and laughed, and he said, "I've got a bridge that I want to sell you in Brooklyn." I talked to him again a couple of years later, and I was like, "You still got that bridge for sale?" Because he was like, "Man, I don't know how you're doing this, but like, you're the only person I've ever heard of that's taking aronia berries and making a fertilizer out of it. Like, I didn't even think that was a thing that was going to be possible. Or you're going to be out of business in like 2 months." And I'm like, "Well, we're here 10 years later, so yeah, it works. It works, and we're here not because of like hardcore marketing or anything like that. We have no marketing department. Me and my business partners are the marketing. Like, we come up with pretty much everything. I think the reason that we're still around and still relevant is because our products work remarkably well, and because we're doing stuff that's way different than stuff that you're going to find in bottles and salt-based fertilizers. Like, you can't ever put what we're doing inside of a bag. No way. It does not work. You lose all of the chemical reduction in that kind of gas phase, So yeah,
I think ferments are the future of nutrition for plants. Really, I was recently at a mycology site where they're doing, basically like some really high tech advanced mycological fermentation to produce some unique compounds. And so I've been using ferments in the garden for a long time, and it's, in my opinion, it's I think going to be the future of sustainable agriculture.
Yeah, and looking specifically at like what are some of the compounds that plants are sensitive to, because it's like, think about it like this: we have at some basic fundamental level, we have these molecules that you can't really break down any further. Like CO2 that's in the air, you can't really break it down any further. If we're talking about sugars, we can oxidize those sugars. If we're talking about organic acids, we can oxidize those organic acids, and the terminal point of that oxidation is going to be CO2 and H2O. But we can't really, like, it doesn't make sense for us to try to figure out how do we break the CO2 molecule apart even further. Sure, carbon monoxide, yeah, but whatever, that's not really relevant for plant metabolism. So if we introduce these very basic materials like CO2, like nitrate, NO3, like sulfate, SO4, these basic building blocks, we watch plants enter them into metabolic processes that are dedicated, and we watch the throughput of that end up in the form of something like an amino acid, for example. If we go a little bit further downstream and say, gosh, I wonder if instead of introducing CO2 to help the plant create an organic acid, I wonder if I can feed the plant an organic acid and then kind of get closer to the point of a terminal metabolite or a dedicated end product. I can introduce that into a plant and watch the plant responds to it. So now this is really interesting because we have this like ultra basic material. We've got one step up, which is maybe a simple organic acid, a dicarboxylic, monocarboxylic, or perhaps a tricarboxylic acid. This is why acetate, malic acid. So acetate, malate, and citrate are of key interest here. Acetate, obviously, for acetyl coenzyme A, and for pretty much everything, even the primary growth hormone in plants, indole-3 acetic acid. IAA is a rooting hormone. It happens to be you know, based on the acetate group itself. And then in the chloroplasts, we see a malate valve specifically whose job it is to export reduction power, because all of that stuff is generated in the chloroplasts and you need to export it out. Otherwise, you end up with overcharging the chloroplasts. There's too much energy coming in relative to what's going out. Sunlight's coming in, chloroplasts are absorbing that energy, photon energy, and converting it into chemical energy. That chemical energy needs to get out as fast as possible, because if there's backflow that fills up, then you start to over-energize the cell, the chloroplast, not the cell. So we have malate valves on the chloroplasts, and then we have citrate valves on the mitochondria too. And they kind of open and close in relation to each other. So the flux of malate leading out into the cytoplasm and subsequently entering the tricarboxylic acid cycle, the TCA cycle in mitochondria, and then coming back out via the citrate valve back into the cytoplasm. And in some cases, like with photorespiration, there's recycling of CO2 that leads back to rubisco. So you have this like beautiful balancing effect that happens with plants where you feed them CO2 gas and you watch the increase in performance happen. You feed them simple organic acids, you watch an increase in performance happen, right? Acetates, malates, citrates, and you keep getting more and more complex with things like kelp extracts. Right now, we're dealing with huge molecules. We're not dealing with simple, low molecular weight organic acids. These are big, chunky, complex, monster behemoth molecules. In some cases, they're 30, 40, 50 carbon atoms long. Like, these are not simple compounds at all. They're remarkably complex, so complex that they even have their own optical properties. If you've ever seen kelp extracts, you may have noticed they're not translucent. They're not clear. There's a little bit of like something's going on with the color. So even the light itself that's coming into the solution is being filtered out by the length of these molecules. So there's something going on here. And as it turns out, these really complex molecules plants are also responding to. We can put kelp extract on our cannabis plants and watch the kelp extract do something really interesting on our cannabis plants. So my specific question is, like, at what point is there a point at which plants stop responding to compounds that are applied externally on them? It's not just the kelp that the plants respond to. Kelp, aquatic plants, and terrestrial plants like cannabis have had separate evolutionary histories for arguably hundreds of millions of years. How is it that there is so much overlap in the logic of metabolism that you can take an extract of a sea plant, apply it to a terrestrial plant, and watch the terrestrial plant do something remarkable with it? That's hundreds of millions of years of evolutionary separation coming back, and one is responding to the other in a pretty remarkable way. So from my perspective, terrestrial plants are really fine-tuned to respond to these things, whatever it is we put on them. So I'm of the opinion that plants will take up pretty much anything that you put on them. If you give them lead, the plants will take up lead. If you give them cadmium, they're going to take up cadmium. If you give them organic acids, they're going to take up organic acids. If you give them anthocyanins, they're going to take up those anthocyanins. If you give them a foliar spray of anthocyanins, they'll absorb those anthocyanins through the foliar spray, because that's what plants do. They're remarkably sensitive to their environment. They absorb everything from the gases in the air to the simple sugars that are being produced by microbes and fungi. The root exudates are feeding the microbes and the fungi, and then in turn the microbes and fungi are producing their own compounds to help feed and accelerate the plant's growth. Same thing in the phyllosphere. Plants are producing certain metabolites that cross the stomata, and microbes are also producing metabolites that get absorbed back into the plants via the same pathway that CO2 gases might. You've got microbes that are producing volatile organic compounds in the soil that actually travel up and enter the stomata from the inner side of the leaves. So basically, this is a system where plants are open to pretty much anything that you can possibly think of putting on them. They will respond to it. They'll respond in a way that maintains a kind of equilibrium. So you have to be careful here and not assume that just because you put something on the plant means it's going to have a fundamentally different reaction. Again, going back to this idea of the roadkill skunk terps can partially be attributed to volatile sulfur compounds, but that doesn't necessarily mean that I put more Epsom salt on my plants and I get roadkill skunk terps. It's not that simple and straightforward. There's equilibriums that are being maintained by plants that you can tap into, but it's not all molecules that will tap into that potential. Those molecules are the ones that tend to interact more with the nucleus within the cells to elicit certain things like the transcription factors that come along and upregulate or downregulate the biosynthesis of certain enzymes, which then lead to the production of certain compounds. You know what I mean? So, yeah.
Stoked. Couple more questions. I don't know how much steam you guys have left, but we've been going for just about 2 hours. I had at least a couple more questions queued up if you wanted to entertain them, Nick.
Yeah, let's do it. I probably have to go in about 5 or 10 minutes, but
okay.
Let's do a couple more questions.
Okay. Here's one from Silent Truth. He's a longtime FCP head, and it's kind of 3 comments that are all going to one. So here he's just kind of stating that he's been using sparingly just the lush bloom and the peak bloom with good results. His next comment was, will there be a 420 sale? I want to try more of the program. I feel like that's a perfect segue into our show for potentially a week from today, maybe next Thursday. And then you want to hit that one before the third one or no?
Yeah. I was just going to say on the note of the 420 sale, we are going to have a 420 sale that starts. Officially, it starts next Friday, the 17th, but for the FCP community, we're actually going to start it on Thursday, the 16th, and hopefully we can do a show next week, in conjunction with that. I'd like to answer questions that people have about products specifically, and just kind of, help people answer any questions they have before, deciding to try our products. With the 420 sale, it's 42% off, so it ends up being the best discount that we offer. So it's a good way for people, and I always encourage, try a quart of some of our products if you don't want to, dedicate yourself to a full gallon or anything like that. So it kind of makes it easier for people to try it. Yeah.
I think that's how I got turned on, was 4 or 5 420s ago. I was like, that's a great deal. I'm going to try it. And I was hooked.
Yeah, I'm definitely going to give it a run and see what it's all about. I'm actually pretty excited about it myself. I've been making my own fertilizers for a better part of a decade, and I'm pretty hesitant to bring new products in, though I do from time to time. I usually use commercial gardens for that experimentation. But I'm actually excited after this conversation, Nick, to get some of your gear and put it to work.
Yeah, I'd love to hear your feedback. And as somebody that's been making their own fertilizers for a while now, I'd love to hear how you think our products look and smell. And Root Anchor in particular is a really interesting one because we use a lot of it's an unalkalized high-fat cacao powder that we're working with a farming cooperative down in South America to source, and it's the cacao butter fraction in there that always really intrigues people that actually know like what the benefit of feeding fatty acids to your plants, specifically as it relates to polyketides and olefolic acid. Root Anchor does something that just like really brings the grease factor out in the flowers. Like it's just the cacao butter, specifically those fatty acids. You're not going to find it in any other fertilizer. I haven't seen anybody else using unalkalized high fat raw cacao. Yeah, and even for people that do it themselves at home, it's a little bit of a unique thing. So I'd be curious to hear your feedback and how it works for you.
Yeah, I've explored the fatty acids in the past and tinkered around with that a bit and found it. I can't think of any commercial fertilizer or even smaller brands that are doing anything like that. That's pretty cool.
And it's pretty much a ceremonial grade cacao. Like I was telling Travis a couple of episodes, maybe a few years ago, like this cacao powder, it's made with a varietal cultivar that was thought to be extinct until the mid 90s, and it was rediscovered. And the cooperative we're working with was one of the first that started, they isolated it, they identified it, and they started planting it on farms, and we've been growing with them. But it has this like really powerful flavonoid profile. Like I can't eat that cacao and then go on a show. Like it's so stimulating. It effectively is a ceremonial grade cacao that we're using, inside of Root Anchor. So I always like to stress to people, if you've never had like real authentic cacao before, like the shit will fuck you up. Like it's strong. It's very potent. It's like a dab straight to the brain. Like it can be very intense for sure.
You got the plants buzzing again. You got them drunk. You got them cacaoed up. I love it.
Yeah. And I mean, yeah, because it's a raw, unalkalized, high-fat cacao powder. It has a very high fraction of cacao butter, but it also has these very unique flavonoids that, to some extent, are kind of rare and exotic in nature. But they have all these wonderful redox active properties that I find. I'm telling you, there's something about this polyketide / olivetolic acid in cacao. Like there's some synergy going on between the fatty acids in the cacao butter and how greasy your flours actually get is kind of what I'm trying to get to. So yeah.
Yeah, we lose a lot of that in the commercial product that's on the shelves right now. It is I've I wouldn't say I've been forced, but I've recently had to uh, go shop at some dispensaries to check out product from certain commercial grows throughout Massachusetts. And man, I'm telling you that like there's some like decent bag appeal out there, but just dude, it's just powdery, dusty, over dried boof. It like the terps are not there. Like it just and it, the that like breaking up your flour and like your fingers being all just like greasy and coated and stick like it just doesn't exist in the commercial market. At least not here. And so I think if you get a couple of producers putting that kind of flour out, that would rock the market.
Yeah, I mean, all of the commercial accounts that we have. I mean, I will say that our products, just due to the nature of what we're doing, using food grade plants that are certified organic, working directly with farmers. We're not buying bottom of the barrel scraps. Like when I mentioned that we're buying, sage from that farmer, she's not giving us like some low grade, scraps of sage that she harvested for otherwise, her apothecary or her herbal business. This is intended for human consumption. It's the highest quality sage that you could ever find for anything you would want to use sage for, or lavender or juniper, or even the cacao that I'm talking about. This isn't something that Hershey's is buying and making dirt cheap chocolate bars out of. This is like ceremonial grade cacao that people who are looking for some kind of like a mystical experience would stumble upon and access this level of quality. So our products end up being more expensive than cheap salts from foreign countries as a direct result of that. But even having said that, like we have commercial producers all across the US, here in Washington, all the way out to Maine. Shout out to the Fractal Boys, doing amazing work. They managed to make our products work, and I will say that the quality is something that consumers can definitely detect. And usually the feedback that I hear revolves around the cleanliness, how smooth the smoke is. There's no harshness. It doesn't scrape the chest. Like it feels remarkably smooth to the point where you almost don't even feel like you took a hit at all. So there's all these like visceral qualities that are associated with good quality cannabis. It's in addition to what you're saying with the specific tactility of it. How does it feel in your hands? Is it really sticky? Like do you have trouble peeling your hands apart? How does it smell? Does it like make your brain tingle right away? There's also these other aspects of the experience during the smoke. Like, do you have a nice aftertaste after you smoke it? And that specific thing about rooted leaf grown flower is something that I've heard more often than not, specifically about the aftertaste. Flower grown with rooted leaf smokes really smooth, burns really well, but the aftertaste is something that people really enjoy because it lingers on the palate, and it's this soft sensation. It's almost like in the aftertaste, it doesn't get heavier. You don't get this like carbon film kind of sensation. It actually feels a little bit more light and airy, like something is dissipating off the surface of your tongue rather than continuing to build up weight on the palate, if that kind of makes sense.
It does very much. And yeah, now I'm even looking more forward to doing a run with your gear.
Yeah, the flavor is next level. I've been calling it flavor expectorant recently, where it just makes you drool certain terms. Like I don't know if that, like how you say like soft and dry. I'm in the total opposite the way I've been putting it recently. I'm like, you want a drool flavor? Take a puff of this.
Yeah, and part of it is like I don't think there's much of a horizontal transfer into the plants. Like we have these compounds we're extracting from sage and juniper, and like if you look at the compounds that we're working with, you're clearly going to find a lot of overlap with cannabis plants too. So they're producing very similar compounds, but it's not like we take a lavender flower and extract the terpenes out of it, and then somehow find a way to put that terpene inside of your cannabis trichome head so that your flower now all of a sudden tastes like lavender. You know what I mean? It's a little bit different than that. There's less of this horizontal transfer thing going on, and more of part of it is a stimulatory thing. Going back to this conversation about farnesene and the farnesene levels that we see with flower grown with rooted leaf, I just feel like part of that is the idea or the notion that FPP, farnesyl pyrophosphate, is kind of an intermediary branching point that's important for cannabis plants to be able to express, specifically as it relates to sesquiterpene biosynthesis. So if you have a plant that produces a large concentration of beta caryophyllene, for instance, which a lot of plants do, I think that's one of the dominant sesquiterpenes. Chances are you can also get that plant to express a huge increase in how much farnesene it accumulates, because part of that is in discussion and revolves around how do these other sesquiterpenes get fed through the biosynthetic pathway from isoprene leading up to a sesquiterpene compound. And then the other part too is maybe at a certain point of metabolic excess, the plant can just afford to accumulate a lot of farnesene. And like it doesn't need to differentiate it further, which is why we see farnesene as one of the things that gets pegged as a little bit higher. Farnesene happens to have a really nice floral aroma to it. I oftentimes attributed it in the past anyways to the jasmine flowers that we were using in resin bloom, because there's a lot of jasmine flower that we're extracting. It's not so much the jasminates, because those are more or less oxidized when they're fresh, but there's conjugated derivatives that remain in the flowers that we're working with, and I attribute a portion of the farnesene presence to the use of jasmine flowers. But I also see the back end biosynthetic logic, like cannabis plants are actively skating around this sesquiterpene pathway anyways. So for them to get a little bit of extra fuel in the bank, in the tank, they're already redlining this process. They're already accumulating a ton of beta caryophyllene and other sesquiterpenes. They've already specialized at that. So to give them a little bit of extra substrate and see that substrate build up isn't an unusual thing. I think if anything, it could be a sign that maybe there's some kind of equilibrium state that's being achieved in the plants. So,
yeah, I think that's one of my favorite things about rooted leaf is that it doesn't necessarily impart a flavor like a generic flavor. Like let's say I grow an orange, a lemon, and a kush, and they all taste like Botanicare or XYZ company. I don't want to throw them under the bus, but that one specifically, I can say kind of has a flavor, and a lot of people will recognize that. Like, oh, this is pure blend. You can kind of just taste it, but I'd say the rooted leaf program only makes the genetics taste more like themselves. I've never seen them kind of come closer together where there's just a homogeneous flavor that comes out of it. If anything, it separates them more. The orange tastes more orange, the lemon's lemon, XYZ. So that's one of my favorite things. You don't get just that stale baseline flavor that a lot of programs kind of tend to lend.
Yeah, and I've never seen any kind of fingerprint as far as a measurable imprint that our products have. Like even with the farnesene, I've seen hundreds, if not thousands, of different terpene results from all across the country, because we've got customers all over the place. And farnesene is always one of those I can sort of like predict, like make sure you measure the amount of farnesene that's in the flowers, just so I can see it too. But usually labs will come back and they'll provide alpha, beta, and cis farnesene. There's 3 different flavors of farnesene that the labs will return to you and say this is what your farnesene content is. And I've never seen the same ratios between all of those. I've seen all 3 of those always expressed in over 90%, maybe over 95% of all the tests that I've seen. They're expressed on a level to where even the analysts might be like, that's pretty cool. Like I typically don't see farnesene levels that high. but the ratios between alpha to beta to cis farnesene, I've never seen overlap between them. I've seen levels of alpha farnesene super high and the other two very low. I've seen alpha very low, just beta high and cis low. Like there's no rhyme or reason to it at all. The only thing that is true seems to be that cannabis plants grown with rooted leaf produce more farnesene that's measurable. And that's pretty much it. And so as a result, yeah, your flowers are going to smell a little bit more floral and smooth. It might remind you of somewhere between apple blossoms and jasmine blossoms. And, you know, it doesn't interfere with any of the skunk or funky armpit kind of gassy aromatics either. If you've ever smelled Peak Bloom, the product itself, if you've ever smelled it, you'll know really quickly that there's nothing about our products that gets in the way of expression of super gassy, very extremely funky. Like you'll smell artichoke and start laughing because it smells like something so foul. Not foul in a bad way, but it smells so good because it's like sweet artichoke. It smells like dates, syrupy. It's like very sweet agave, but it has this distinct like raspy kind of like gassy, funky, earthy artichoke, aronia berry. Like most people laugh because it's such a complex aroma. They're like, "Whoa, this is like the best smelling dead skunk in the world," kind of thing, you know.
What do you guys think? Should we wrap this one up until next time?
Yeah, I should probably get going. Got to make some dinner here.
Let you settle in, get fed. Yeah, there's a bunch of great comments and questions in the chat. I'm going to try to write them down maybe next week. Ooh, echo. Mr. Toad specifically had some interesting questions, but we'll be able to get to them next time. And if anyone has any new ones, but I heard you coming in there, Josh. Anything you wanted to say before we get off?
I just wanted to say thanks, Nick, both of you guys. This was very informative, and I think it was you know, a good opportunity to talk about some aspects of plant biology and the importance of electron chain transport and carbon in cannabis cultivation and chemistry that's not talked about enough. So it was a pleasure to be a part of it. Thank you both.
Yeah, thank you. It was great to meet you, Josh. I hope we have more conversations in the future. And Travis, always great to talk to you. Hopefully, you know, we'll make this more of a regular thing too, because it's good to bring the whole community along this learning process for everybody. Sugar phosphates are not something that are really talked about in the context of fertilizer application, but you know, Josh is an incredibly intelligent guy. If you asked him, he'd be able to tell you all this wonderful information and the benefits that it has. So for me, this whole thing is about just like bridging the gap between what do people know versus what are we actually doing. You know, like let's just get to a point where we're just more centralized on like the latest and greatest things that we know we're actually capable of acting on in the garden. We can use these tools effectively to steer the kinds of results that we want to see with our crops. Overall, better health, better plant expression, decreased reliance on synthetic pesticides and fungicides. I'm really not a big fan of either one of those categories of compounds to begin with, but all of these things can kind of get wrapped up in the same continuous dialogue. So I look forward to extending this in the future with hopefully Josh and others and Travis and Peter and everybody else that tunes in and watches this.
Yeah, hopefully we'll be able to pick up next week and keep the ball rolling.
Yeah, tune in next week. We will launch the 420 coupon code as well. So for those of you who are interested in trying our products, I can activate the coupon code right before we go live. And if you guys want to ask any questions about how our products work, what they do, what's inside of them, I'm an open book. You can ask me whatever you want, as long as it's not process related of how I'm actually making it. I'll probably answer it. You know, if you want to know something about the plants that we work with, or something cool that the plants do, or if you have any use specific questions, next week would be the time for that.
Awesome. Pleasure and a privilege to talk to you, gentlemen. And as always, an honor to be a small part of FCP. I believe there is another show going right now. James and Eric are talking about a soil company, I believe. So anyone who's watching, still, you can hop over there and keep the education rolling. And until next time, thanks so much, guys.
Thanks. See you, Travis. Thanks, Nick.
Later.